We then went from there and looked at patients with ALS and asked the question, are there regulatory T cell alterations compared to normal controlled people? And we found, much to our surprise, that the regulatory T cells were decreased in number and decreased in their neuroprotective functions. As a result of that, we then said, well, this may be a meaningful, effective therapy. Why don't we take these T cells out of patients and expand them outside the body? And if we have to reprogram them to make them normal and healthy, we could do that. But when we took them out of the body, it turned out we didn't have to reprogram them. By removing them from a toxic inflammatory milieu, the regulatory T cells became normal as we expanded them in the presence of IL-2 and rapamycin. Very, very exciting, because it said to us, we can take bad Tregs, turn them into good guys, and then infuse them into patients. So we made a request to the FDA, who approved a small pilot experiment in which we could give the patient's own regulatory T cells that were toxic, remove them, expand them, they became normal, and give them back to the patients. We did this in 3 patients with ALS, and to our great surprise and pleasure, the patients stopped progressing. Their ALS stopped progressing.